Good afternoon from me as well. It's an absolute pleasure to be here in what I believe might be the final lecture of the afternoon, and I'm really grateful to the organizers for inviting me across from Maastricht University campus, which, as you know, is really next door to here, but particularly also to make the neuroimaging the final topic of the day. And so clearly the organizers are convinced that it's a good idea to consider neuroimaging in psychiatry. I'm not sure whether everyone in this audience is, especially at this time of day and stage of the conference, and I'm trying to convince you that indeed neuroimaging, particularly functional imaging, might be something that's relevant more broadly for psychiatry. Although it is probably fair to say that some of the hopes and promises, expectations people had about 20, 25 years ago when I myself started in psychiatric neuroscience about neuroimaging becoming almost a bedside tool haven't been fulfilled. And indeed, in clinical practice, probably a lot of what we still do with neuroimaging is rule out organic diagnoses. We might even hand over patients once neuroimaging has brought up something to neurologists or other specialties, although we may still be involved in some way. But generally, neuroimaging is not considered to be a positive diagnostic, or give positive diagnostic signs in psychiatry. And I'd like to spend the 20 or so minutes now to consider jointly with you whether it is likely and also would be positive and desirable that that might change. Starting from diagnosis, are there ways of using imaging, particularly functional imaging, beyond the kind of mechanistic investigation about we just heard in the previous presentation, also to diagnose diseases, syndromes, presence of symptoms, and then moving on to uses of neuroimaging in brain-based communication, and finally also prospects for neuroimaging-based therapies. So the first topic is around the buzzword of brain reading. The idea of inferring mental states and tensions from brain activation patterns, and that could be fMRI, functional MRI, which looks at the vascular correlates, oxidation correlates of neural activities, but could also involve electrophysiological techniques like EEG or MEG we just heard about, complemented perhaps even with the peripheral physiology. And there are, in very few cases also, more on the neurological side, invasive multi-electrode recordings. So if we wanted to build up an argument, which I would say is actually not my argument, but one could build up an argument saying, okay, we lack objective markers of mental states. Well, that's almost philosophically true if you think that mental states are defined by the first-person perspective, and that is, of course, one of the essential limitations of psychiatry, and perhaps will always be that we have no direct access to the patient's inner world. And again, that's both a neuroscientific, philosophical, ethical debate. Do we want to have more objective neuroscientific access to these mental states? I think proof of diagnosis or uses to detect malingering are neither desirable nor possible. But I think a more positive and productive way might be to look at neuroimaging to understand more about mental states and then perhaps try to use them also for modification in my last topic, which will be neurofeedback. In neurology, there's perhaps a more established area of brain reading, mainly in the context of brain-computer interfaces, control and diagnostic applications. I'll come back to this in a moment. So let's just start with a relatively common and central symptom in psychiatry, hallucinations. Early work from our group showed that indeed it is possible to map out neural correlates of auditory verbal hallucination. This is the activation pattern in the primary auditory cortex during verbal stimulation, external hearing of external voices. This is the hallucination correlate. So we can detect correlates at individual levels of symptoms. We can even go slightly further, looking at content slightly more. This was a case study of a patient with visual hallucinations. This is a flattened representation of the cortex. with the functional clusters indicated in color. This is the occipital, the posterior part of the brain, occipital cortex. And the areas highlighted here are the areas activated during hallucinations in specialized visual areas like the occipital face area, extrastriate body area, parahippocampal place area, and they mapped onto the content reported by the patient about the hallucinations of people being in particular landscapes. So to a certain extent, we can go beyond mapping out a symptom into mapping out content. But the reverse inference of inferring a mental state from these brain activation patterns will still require many, many more steps. And I'll tell you about the difficulties of that in my next few slides. Because one of the things we did afterwards was compare hallucinations with imagery. And this is the group activation pattern, for hallucinations. This would be for imagery, coronal view of the brain to sagittal views, showing essentially inferior frontal areas, showing the superior temporal sulcus. And this frontotemporal activation pattern was actually very similar, including activation in the human voice-specific area and superior temporal cortex, which was happening both during when people were reporting hallucinations and when people were instructed to imagine voices. So yes, the phenomenal experience of hearing a voice we could map out, but not really distinguish between hallucinations and imagery. In this case, actually, it was the proband group, the voice hearers, when the actual patient was a group of non-clinical voice hearers. And about the same time, a group here from the Netherlands, and of course we're talking about Dutch glory, so I'm trying to highlight very important work from the Netherlands, was not for myself, but a very important work here that was done. Essentially the heading sums it up. Auditory hallucinations elicit similar brain activation in psychotic and non-psychotic individuals. Now if you consider clinically, phenomenologically as well, particularly clinically, what a huge gulf there is between people who suffer from the voices and people who report them but don't suffer from them, it's immediately clear that there is a limitation. Yes, we can map out correlates of hallucinations, but we can't really distinguish between clinical and non-clinical hallucinators, whereas we can't distinguish between imagery and hallucinations. So that, I think, is important to consider. I think it's of great interest, possibly even at the individual level, possibly even something to discuss with the patient, what we know about the process going on in their brain, but a major limitation for any actual diagnostic use, especially if you wanted to disprove phenomena experienced by patients. So I don't think that will ever be a potential, and as I said, certainly also not a desirable application. Moving on, so what perhaps we can do diagnostically with these kinds of techniques, though, and I'm moving slightly more into the area of the sort of borderland between psychiatry and neurology here, which sort of reflects my own personal interest in the clinical neuropsychiatry. And if you look at this taxonomy of disorders of consciousness, there are actually a few areas where neuroimaging, neurophysiology with functional signals can help us differentiate. One example would be epileptic and dissociative states, for example, where there is an established use of EEG, which is also more broadly a functional imaging or neurophysiological technique. But another example would be, and I'm referring particularly to very important work from Liège here, to distinguish between different levels of disturbance of consciousness, functional imaging to differentiate between vegetative state and minimally conscious state, for example, that many of you will have heard about. It was really also big in the media about 8 years or so ago. And this is the example where essentially people who were diagnosed with a persistent vegetative state were in the scanner asked to imagine tennis movements. And it was found in this group of about 50 or so patients that 5 patients actually, although they couldn't actively, conventionally communicate, showed the corresponding activation patterns here in higher motor areas for the motor imagery and here in parahippocampal area for scene imagery. So these are patterns that would indicate that these people actually had preserved understanding and weren't suffering from a complete try and get back. A complete lack of awareness, but we're somewhere further up on this spectrum. So this is obviously a very specialized application, but one that gives you an idea how we can clinically already, at this moment in time, try and explore the use of these functional imaging techniques to infer mental states, in this case, in cases where there is no other obvious way of communicating. Moving on with the communication theme, in the sort of spirit or concept of brain-computer interface, which I've indicated here, which is essentially a circuit where we record brain information, transform it into algorithms that can then control an output like a robotic arm, but also a computer cursor. So it can then be used for people who don't have active motor output to communicate, and I'm referring here to the cases of locked-in patients. Now here we're not talking about a disorder of consciousness awareness preserved, but disorder of communication. And there again, ideas of reading out intention or meaning from brain activation is of considerable interest in terms of potential communication techniques, and they're invasive. There's, of course, as many of you will know, important work with invasive brain-computer interfaces going on in Utrecht. And the example I wanted to show you here is from across the road from the imaging center here in Maastricht, from Bettina Sorger, is an fMRI-based communication interface where essentially people learned to control a speller. There were 27 cases, 27 potential sort of instances. I think that was just fine, of course, for the alphabet. 3 different mental tasks: motor imagery, mental calculation, inner speech, which you can all easily learn, and which can be differentiated quite nicely in terms of their brain activation patterns, with 3 different onsets, 3 different durations. So in a minute, you can determine which of these letters you want to pick, and then it takes some time, but that's the case for all of these brain-based communication interfaces. This person here in the scanner had to spell the name Victor, and the letter they needed to select was R. And the R here on the grid that they saw could be selected by inner speech for a certain duration of time and with a certain onset. So this person was imagining was just reciting Shakespeare to themselves, which activated the right image, right part of the brain at the right time. So this is a way, and you could do similar things with other readouts of brain activity, as I said, invasive or non-invasive. So with these kinds of techniques, we can actually show specificity of brain activation, and this can be used for communication, clinical application then, potentially in people who have no other means of communicating. So this, I spoke about brain imaging, brain reading for quasi-diagnostic purposes, spoke about using this in a brain-computer interface context for communication. And my last topic will still be in the area of brain-computer interfacing. In this case specifically, we were using fMRI, again, the functional MRI signal, but many other groups would use EEG in this context of using the brain-computer interface in a kind of closed loop to teach patients to self-regulate particular patterns of brain activity. And there are multiple clinical applications. In fact, there's even a field of practitioners, especially in the area of EEG neurofeedback, and I'm going to focus my own research is more in the area of fMRI-based neurofeedback. I'm going to give you a few clinical examples. So the basic idea is if we think either that a particular pattern of brain activation might be faulty in patients, we could try and help them self-regulate that activation through this training process. So they see in real time the brain activation while they're in the scanner, and they're trying to change it. And then they see from the feedback, for example, the thermometer here, whether they're successful or not. You could, though, also think that, because it's actually difficult to show in individual cases that a brain activation pattern is faulty, and also, for many diseases, we don't know that yet, whether that's the case and where. You might also think that you'd like to reinforce a particular pattern of brain activation, no matter whether there's good evidence for it being faulty, in a kind of an exercise framework. So you might think that training activation in particular emotion areas for some patients with some psychiatric syndromes might actually be beneficial as a kind of reinforced imagery process. And that is kind of the pattern that we've been pursuing in depression, which I wanted to tell you. But first, to show you that it's actually this is an actual field of research interest. Hasn't entered the clinic yet. EEG neurofeedback has to a certain extent, particularly in ADHD. But there's a huge and increasing interest. So this is just cited from the British press from a couple of years ago. Tests raise hopes for radical new therapy for phobias and PTSD. Researchers able to reduce anxieties without asking people to think about them consciously, making it more appealing than current therapies. Now, I'm sure that's food for thought here for this audience, and quite a challenge, in fact. But if you think about it as a potential complementary avenue to treatment, or perhaps to lead people patients into certain treatment, it might be of interest. This referred to a study, actually not in patients, an experimental study by a Japanese group, which uses conditioning paradigms based on brain activation patterns related to aversive stimuli. So just one example of the sort of wider interest in this field. So my two clinical examples will be on depression and addiction, which are areas I'm involved in, and there's also a strong ongoing collaboration, long-standing in Maastricht. This slide just highlights some of the areas that one could target potentially in neurofeedback in depression, and you'll notice there is some overlap conceptually as well with other brain stimulation techniques, although neurofeedback, of course, is non-invasive. We did a study which just recently completed, where essentially in a controlled trial, we had 5 sessions of neurofeedback. What we asked patients to do is, after we'd localized areas that are functionally responsive to positive affective stimuli, we asked them to upregulate the activation in these areas, and that's what they practiced over 5 sessions for about half an hour each, and we collected depression scales pre and post. And half the patients were randomized to this particular exercise of practicing positive emotional imageries with neural reinforcement. The other group, and if you remember the slides I showed you about the PPA from parahippocampal place area from the functional imaging study in persistent vegetative state, they activated the parahippocampal place area with scene imagery, which was supposed to be a sort of neutral control intervention. And the next slide shows that actually these were the target areas, parahippocampal place area, and sort of emotion areas in the insular paralimbic areas in the active group with the. So that's what people were activating over these 5 sessions, and they managed to do that. And these are the clinical outcomes, which we could spend a lot of time discussing, and perhaps those who are interested, we could do that later. And essentially, the bottom line is there was an improvement, about 40% on the Hamilton scale, but in both groups, and which could, of course, mean many things in a controlled trial, including placebo effects, regression to the mean, but also effects in the control group, which could all be discussed. There was a recent trial with a relatively similar paradigm with upregulation of the amygdala with positive autobiographical memories, which had a slightly different control condition and actually did show a clinical improvement in the active group similar to ours, but no improvement in their control group. So I would say it is probably worth pursuing this area in depression, although the jury is still out whether it'll ultimately be an efficacious treatment. And just briefly on my last topic, addiction. There's a different way of approaching in our group addiction with neurofeedback, not by asking people to imagine, but just sort of looking at the thermometer or rather abstract stimulus. We use the activation produced by addiction-related cues in the ventral striatum, And we use that, we feed that back by showing the different sizes of alcohol-related cues. So patients will see the abstinent patients with alcohol dependence saw these kinds of stimuli, and their task was to make them smaller by downregulating activation in the ventral striatum. And we did similar work in relation to overeating as well. So this is still ongoing. I can't give you the results yet for this, but certainly another potential way of using neural activation to teach patients self-regulate the incentive salience of motivational stimuli. So this is something we're pursuing at the moment in a large European project, where we're looking at alcohol dependence, and other groups are looking at anxiety disorders, autism, and eating disorders. This is just the map of this consortium. We call it the Brain Train Consortium. And in fact, we'll have a clinical stakeholder workshop to discuss these things in more detail, which we'll advertise soon in September, which may be of interest to some of you. And just to show you here that this is an active field of investigation, even just only fMRI-based neurofeedback trials across the world, about 40 or so I found and those are just the ones registered on clinicaltrials.gov. So a very active field in both Europe and the US at the moment. And if you add EEG neurofeedback to that, the picture would be much larger. So this is probably something you'll hear more about. It's not definitive yet in terms of clinical application, but certainly an area where quite a few research groups are investing in at the moment. Just so to summarize, I spoke about symptom mapping, which can be done, and quite specifically, you can find correlates of hallucinations, cravings, for example. I didn't go into the details here. Well, you can find them. The specificity is the problem. They're anatomically specific, so you can find the corresponding areas, but as I showed you, it's hard to differentiate that from related phenomena like imagery. Brain reading, I gave you the explanation or the example of disorders of consciousness, the moment very specific case. And I think we need to approve this topic very carefully, but potentially very interesting applications. Brain-based communication, perhaps less directly related to classical psychiatry, more in neurology, and neurofeedback, which could be potentially an attractive tool across quite a range of mental and behavioral disorders. And I guess, again, this is just a thank you to our Brain Train Consortium and a few funders. I assume there won't be time for questions now, but I'll hang around afterwards if anyone wants to ask me in more detail. Thank you very much.